talk about device-based therapeutics, which is really an interesting area, as I've spent most of my career around more small molecule-based therapeutics. So it's been exciting for me to move into a slightly different area and be able to combine the idea of a device with a therapeutic. And so our company EnClear Therapies is focused initially on CNS diseases, but I'll tell you how it potentially can be applied to other diseases as well. So the cerebral spinal fluid, which I have in this diagram in the blue, is the liquid that surrounds your brain and flows up and down your spinal cord. And it's really been largely ignored for all intents and purposes, although it has been used a little bit as a diagnostic, and it's really becoming more and more apparent that it could be used as, quote, a window into the brain. So instead, people don't like to give pieces of their brain away. It can be a way to sort of get a sense of what's going on in your brain without, too much invasion. And as we age, like a lot of our systems and our organs, we tend to accumulate toxic proteins in our spinal fluid. And then in certain diseases, also you see toxic proteins accumulating, and I'm going to talk more about these. And you can take CSF from patients from certain diseases, and if you take their CSF and you apply it to cells, or you create or to animals, you can get toxicity in cells, and you can create deficits in the animals similar to human disease. So it's believed that this accumulation of these toxic proteins isn't something that's particularly good for you. So EnClear Therapies has been developing a device to basically clean CSF of these toxic proteins, and with the hope that we can slow or stop the progression of disease. Also, the device that we have really gives unique access to both the brain and the spinal cord. And so it will allow, as I'll go into more detail, ability to survey the brain, be able to apply drugs directly to the brain, as well as be able to sample over periods of time, if needed, the spinal fluid to get a sense of whether to look at changes that may be happening. So really, cerebral spinal fluid is the key to having good health. And so just like blood, it's something that can be monitored and has been found to change over time and change with certain diseases. So its purpose is it's the most direct connection to the central nervous system. As I said, it's a fluid that continuously flows in the ventricles within the brain and around the surface of the brain and spinal cord, and really serves three primary functions. First, it serves as a shock absorber. So, if you bang your head, it's a good thing to have. It helps protect the nervous system from that shock and from damage that can ensue. Of course, if you really bang your head, there's not a lot that can prevent some damage, but it helps with small insults to your head. Also, it circulates nutrients and chemicals filtered from the blood, and this is actually an area that has increasing interest, from scientists in terms of what are those nutrients, how do those nutrients change, how do they control, how do the release of them and chemicals such as hormones, for example, are known to control certain functions as part of the release into the CSF. And then the third purpose is really to remove waste products from the brain. So it's a way that cells that are getting rid of toxic proteins or misfolded proteins or proteins that aren't needed anymore. It's a way of them, those cells getting rid of those products, and it goes into the CSF and can be further broken down and whisked away through the circulation system. So it serves really these three functions. So what EnClear has done is they've developed a device that basically connects to your circulation of your CSF via a cartridge if we want to treat the CSF versus a pump, so we can actively pump CSF through the system at a faster rate or the same rate at which it normally is being circulated. And this way it can either deliver therapeutics to specific areas of the brain that'll go into more detail. It can target to remove certain toxins, as I had spoken about. You can target certain toxic proteins that are associated with disease progression. And then also for detection and measuring a disease potentially sooner by identifying biomarkers, early on in the disease, and can be more reliable than, for example, taking blood samples. So as we age, as I said, a lot of our systems and our organs start declining, and also in a number of neurodegenerative diseases and other diseases as well. There's a lot of benefits of youth. So those of you who are listening who are young, more power to you, because really our cells have a natural defense mechanism against proteins that become misfolded or somehow are faulty. Our cells are really good when we're especially young to get rid of these proteins. But as we get older, and if you have specific genetic mutations or combination of genetic mutations, these processes decrease in their effectiveness. And this results in, and probably can explain a lot of why neurodegenerative diseases as a class of diseases often appears later in life, because you may have had some faulty ability to clear these proteins, but as you get older, your ability becomes less efficient. And so you can get this accumulation of toxic proteins. And in this image, I have arrows that are pointing to 3 cells. And this is stained with an antibody to a protein called TDP43. This protein in diseases gets mislocalized. It's normally a nuclear protein because it binds to DNA and RNA as part of its function, but it becomes mislocalized to the cytoplasm, and there it accumulates and forms these sort of toxic aggregates. This is just one example in the middle panel, and the last panel that I just show lists a number of diseases. For example, so TDP43 is found to be mislocalized and aggregated in amyotrophic lateral sclerosis, ALS, as well as there's other proteins that are associated that become toxic. Also progressive supranuclear palsy, PSP. So Alzheimer's disease also has a number of proteins, both in genetic forms and sporadic non-genetic forms of Alzheimer's. Parkinson's and frontal temporal diseases also have proteins that are known to accumulate this way. So it seems to be a broad phenomenon that is a lot of scientists have been looking at various strategies to try to reduce this toxic accumulation of these proteins. So I'm going to specifically talk about ALS, Alzheimer's disease. Amyotrophic lateral sclerosis. It's a terrible disease. There's no treatment options. Patients basically get diagnosed early in the disease with muscle weakness, often some slurred speech, muscle cramps. These are very early symptoms. These symptoms continue, and usually diagnosis happens around 1 year after the initial symptoms. And patients are often wheelchair bound by year 2. Basically, they lose the ability to use their limbs and get this progressive weakness. Ventilator is often required as they have more and more trouble breathing and swallowing. And often by year 3, they're bedridden, and usually death occurs somewhere between year 3 and 5. So a patient may start out mobile and then, relatively quickly end up in a wheelchair. So it's a very devastating disease. Really, time becomes most important for these patients. And so even being able to delay the time that they have to be in a wheelchair delay the time that they require a ventilator. This is really valuable time and is very impactful for these patients. So the device that we're developing, I have a timeline that we're on right now. We plan to be first in human next year, which is quite exciting. And by 2023, we'll have a pivotal trial that will really determine, the true efficacy of our approach. And EFS stands for Early Feasibility Study for those who may not be acquainted with sort of the device world. So we believe that targeting these toxic proteins can slow disease progression. And there's some literature that supports it. I've just listed a few references associated with it and made a few points. We know that reducing TDP-43 toxic protein burden prevents the cytoplasmic inclusion. So if you can reduce the levels, you can reduce the cytoplasmic inclusions, which are aggregated forms of the protein, and thereby rescue the neurotoxicity. And these are both hallmarks of the disease. Also, if you remove toxic proteins from the CSF before the clinical symptoms in an animal model, you can also prevent the ALS, the disease itself in an animal model. And then last is if you clear the TDP-43 inclusions or stress granules, and these are the things that get formed in the cytoplasmic part of the cell, in animal models, you can slow the disease progression and prolong survival. So this is really what our target is by removing these toxic proteins. We hope that we'll slow or halt the progression of the disease. So here's an image of our, another image of our device. So we can basically circulate, very similar to kidney dialysis. We can remove and circulate the CSF and put it through via a pump and a cartridge to modify it and then return it to the patient. And so basically, as a closed system, be able to circulate and filter out the toxic proteins from the CSF of patients. So the idea is that you have a patient with ALS and their disease results in the generation of these toxic proteins. I've listed a couple of them here. These proteins are going to accumulate in the brain as well as the CSF. And through our method, we can remove those toxic proteins, return the CSF, and thereby lower the levels of the toxic protein for the patients. So here's some of the proof of concept studies that we've done. First of all, you want to make sure that by removing the CSF, treating it, and giving it back to the patient, that you're not going to make them worse. So in this, on the left-hand side, this graph is just, that shows 3 bars. The first bar is just CSF. The middle bar is CSF treated with one of our treatments. And the third bar is a separate treatment. And so you can see that on these motor neurons that have been differentiated from iPS induced pluripotent stem cells, that there's really no toxicity that we see in terms of our treatment. Also, in vitro, we've looked at removal of other proteins known to be toxic. And then we've also done some proteomic studies to look at effects of treating CSF and which proteins are affected by our treatments. In vivo, we've done some small animal studies with rats where we've given either over 7 days or 20 days the treated CSF passed through our system, delivered via osmotic mini pumps, and not seen any effects on the health of the animals. Also, we've done a rabbit study, and we've had to go to sheep really to look at an animal model that we can do the full circulation. So we can put a very similar system as we'll use in people, in sheep, and be able to circulate in the sheep their CSF. And we've done this for 30 hours totally, so 3 10-hour blocks of time without producing any toxicity in vivo. And these studies continue on looking in more depth at some of neuropathology to make sure that we're not producing anything toxic, as we've had our discussions with the FDA. So other indications. So as I said, we're initially focusing on ALS, but this has applications to other neurodegenerative diseases, as I've listed here: Parkinson's, Alzheimer's, tauopathies, Huntington's, and spinal muscular atrophy. Also, in terms of cancer, CNS tumors, CNS lymphomas, meningeal tumors, these are all potential targets for us. There's some rare inherited diseases like lysosomal storage diseases that we could have an impact on, as well as autoimmune diseases that are listed here. For example, in autoimmune diseases, doctors now will sample your CSF to find elevated levels of immunoglobulins. So by reducing the immunoglobulins that end up in the CSF, this would be another potential application. And then additional indications around head trauma, CNS infections and stroke. So these are all sort of all the things that are in front of us. And from this list, we've been putting together a pipeline of indications to apply this therapeutic device to. So the next part of my talk, I'm going to talk about how we've looked at using the system for drug delivery to the CNS. So this is a model, and the solid line here is what happens when you give a drug repeatedly over time. So it's just the free drug kinetics. So you see the increase in the levels of the drug, then a decrease as the drug is being taken up, and then in the next dosage you'll see another increase and then another decrease. And the problem with this is that you can see it's easy to go into the toxic range. You have to make sure that the maximum level of drug that you're seeing is below this toxic range. But then if you don't give enough of the drug, then you're in the subtherapeutic range. So the problem really is, how do you get a steady level of drug that stays in the therapeutic range? And so through our device, using a pump to control and the rate at which you infuse the drugs, we can deliver so that we can get a concentration of the drug that stays within the therapeutic range. So a really exciting area of new therapeutics today is around large molecules like AAV, antisense oligos, siRNA. But when you're applying these to the nervous system, it's really a problem in terms of delivering them so that you can get the concentrations you need and the distribution you want. And then also monitoring, being able to monitor the levels as well can be a challenge. So first of all, one challenge is just penetrating the blood-brain barrier itself. So with our system, we're able to get around that, and basically we're applying it directly to the central nervous system. The other is, so typically these molecules now are given intrathecally into the lumbar region or the base of your spinal cord region, and then you want them to then now go up to your brain. So it's really an uphill battle. The highest concentration is going to be where the drug is applied, but if you want to get it higher up and into the brain, it can be more challenging. And then also it's harder to control sort of dwell time and absorption time, just giving a single injection. And then also it can be painful and cumbersome if people need repeated injections, but we have implantable ports in our system that allow access, repeated access if necessary. So here's an example from the literature where an intrathecal bolus injection of a GLUR1 antisense oligonucleotide was given. So this was given in the lumbar region, and you can see in the dark blue on the far right, the lumbar spinal cord, as a result, has the highest levels of this drug. Next is the thoracic spinal cord, which is the mid region of your back, and then you get decreasing levels now in the cervical region, which is the top part of your spinal cord. And then by the time it comes to your cortex, you have even lower levels. So you get this very strong asymmetry in the distribution of this antisense oligonucleotide, as demonstrated in this figure. So we've done some initial studies with the drug methotrexate. So this is a cancer drug. It does not cross the blood-brain barrier. It's very commonly used in cancer therapeutics. It's been around for a long time, so a lot is known about it. So we thought it would be a good candidate to initially look at applying it with our system, because in the terms of any cancers in the brain, this could be a useful drug if you could target it and get concentrations that you need without exceeding toxic levels. So this is an initial experiment we did, and we wanted to compare initially just if we gave it into the lumbar, similar region as what's given when you do intrathecal injections of, for example, that antisense oligonucleotide. In the far right side, you see that in the lumbar region, as well as the cervical region, are very similar levels. So even though we've applied it in the lumbar region, we're actually getting good concentrations also on the cervical level. And that's translating into the rest of the brain as well. And you can see even in deep structures of the brain, you're getting decent levels of the methotrexate with our system. So, so really we think that we can overcome a number of delivery barriers. You know, one is access. So being able to access the brain for biologics, gene therapies, other large molecules, as well as small molecules that don't cross the blood-brain barrier easily. Our system could be a way to deliver these molecules more effectively. Pharmacokinetics, so based on drugs' half-lives and toxicity limiting concentrations, we believe we can get steadier concentrations of drug without worrying about and being able to control the more toxic levels. And then distribution. So a lot of times, you know, you get uneven distribution as in that publication. So it's possible that we can deliver to specific and get concentrated levels in specific areas of the brain based on how we deliver and how we infuse the drug and control the circulation. So the last is just a little bit about our company. We did a Series A financing earlier this year before COVID. That was good. We have completed some in vitro safety and efficacy studies for a couple of different disease states. And as I said, we're focusing initially on ALS. We've developed an intellectual property portfolio and completed some of the in vivo animal studies that are going to be required for filing with the FDA. And we're on track for putting in our first in human studies the first half of next year. And on the right-hand side, I've listed some of our venture capitalists support. In terms of our team, here's the management team. As you can see, several people come from Medtronic, so they have a lot of experience in terms of devices. I'm on the therapeutic side, but I have a great team of advisors in terms of the clinical side, as well as in terms of the assays that we need and developing biomarkers, et cetera. And then this is partnerships and collaborations. So we have a number of partnerships with clinical centers. Our first clinical trial is going to be done at the Healy Center. We have companies we're working with for R&D manufacturing. our company is all virtual, so we rely on these wonderful partners to do all of our work and help us be our hands, basically. And then we have a collaboration with Therapeutics, located in the Netherlands, for their Antisense oligonucleotide, looking at how our system can deliver better um, than their current regimen they're thinking about. And then at UMass Medical School, we're working with a professor there. And then, of course, patient advocacy groups are always important to interact with. So we've been having discussions with all of the ALS foundations. So just highlights, I think cerebral spinal fluid is becoming, people are understanding it more and more as a really useful window into the brain. We accumulate toxic proteins in our CSF as we age and as well as in diseases. And that's been shown both in cells as well as animal models. We have a device, therapeutic device that we believe will help slow or stop the progression of disease. And also will be extremely useful in terms of being able to effectively deliver therapeutics into the brain and spinal cord. And we are, lastly, we are open to collaboration, new collaborations and partnerships. So please don't hesitate to contact me if you want to talk some more. And there's my email address, my first initial, last name at EnClear Therapies, and I'd be happy to take any questions.
There are a few questions that have been posted.
Great.
Yeah, so
Sam, you were on mute, so you can take it from here.
Yeah, there are a couple questions for you, Marcie, and I'll start with Hugo, CEO of PhenoSwitch. He's asking one general question. We have been doing CSF research for a while. One of the main problems is plasma blood contamination during collection. How do you tackle this problem?
Yeah, so I think the idea is that if you can put the catheters effectively into the intrathecal space and into the ventricle, that you can avoid that. And we, as I said, we use ports that can help that as well. There are ways to test for that as well, as I'm sure he's aware of too. But it's something we'll definitely monitor.
Wonderful. And there's another question from Larry Hardy. The first part of his question is exciting talk. Given the unmet needs for the effective ALS treatment, what happens to toxic proteins after they're filtered out of CSF? Must have an external filter cartridge be replaced periodically? What's the capacity of the filter?
Right, so we've been looking at all of those things, and it's easy to switch out the cartridge. And so we've done, to date, what we've done is we've done some modeling to predict, how the size of the cartridge, the effectiveness of it, so that we can determine that. But it's going to be some work that we'll be doing also in our initial human studies.
Well, and the second part of his question is, can non-invasive imaging methods be used to monitor CNS concentrations during delivery of proteins or nucleic acids into the brains of humans?
Yeah, I mean, there are imaging agents that are being developed and have been developed. I'm not sure if that's what he's referring to, but there are a couple things. I mean, you can do sampling of the CSF as a way of looking at concentrations, but there are some imaging, fairly expensive imaging methods as well.
Is there any other questions for Marcie that she can take?
I have one final question, which is, could you briefly describe the envisioned patient experience, how they would be using it, how long they would be?
Yeah, so what we think will happen, it'll be something that patients will need periodically. But basically there will be implantable components. Initially anyway, there'll be implantable components, and maybe in the future it'll be totally implantable, but it'll be implantable components where similar to kidney dialysis, where you go to a center to get your treatment.
Okay, thank you.
Great. Well, thank you very much for the invitation and the chance to speak.